All right, let's continue with section 2 of chapter 15. We're talking about making transgenic organisms, in particular moving human genes into bacteria. And we saw how we use plasmids to do this, and that the process is not 100% efficient, so we engineer our plasmids to have genetic markers, that is, these antibiotic resistance genes. So, for example, you'll be growing your bacteria on these petri dishes, which are about, I don't know, 3-4 inches across, and you'll have this whole mass of bacteria growing on there. But what you find after you expose it to the antibiotic, you'll find particular clusters of bacteria that survive. The rest of them are killed. So those are the clusters of bacteria that have successfully taken up this gene, and those are the bacteria you can use to create your colonies that are going to produce human growth hormone or insulin, for example. All right, so this allows us again to create what we call a transgenic organism, an organism that has mostly its own genes, but it can have genes from other species spliced into it. We saw that this is done with bacteria. It can be done with plants. With plants, what you do is we use a bacterium called Agrobacterium tumefaciens, and this is a bacteria that normally induces these tumor-like growths in plants. They basically infect the plant and they transfer DNA to the plant cells and cause these growths. Well, what we can do is engineer this bacterium such that it doesn't cause the cancerous growth. We can remove those genes, but we can use plasmids to splice in useful genes that we want to get into that plant. What we do is we take masses of the plant cell, masses of cells from the plant, we expose them to the bacteria, and we can then take those clusters of plant cells that have been exposed to the bacterium, and you can do what's called tissue culture, which is commonly done with plants. Plants are much more easily to manipulate than animals. You can take sort of a piece of leaf or a root from a plant, and under the right conditions, you can get it to grow a whole new plant, whereas with animals, you typically can't take a piece of an arm or a leg and grow a whole new animal. It doesn't work. But it's much more easily done in plants. So you can take these plant groupings and you can grow them in tissue culture, and if the transfer of genes was successful, you'll end up with plants that now have incorporated these genes that have come from somewhere else. This has been done to create genetically modified crops like corn and soybeans and cotton that have incorporated genes from completely different species into them. One of the more common ones is what are known as Roundup Ready soybeans. You may have heard of Roundup. It's a herbicide, a weed killer. And there are soybeans that have been engineered to contain a gene which came from a different type of plant that gives them resistance to the Roundup. So you can spray this herbicide on your field. It'll kill all the weeds, but it won't kill your soybean plants. In fact, I believe I've read that upwards of 90% of the soybeans grown in this country now are this type of soybean, transgenic soybeans. Animals are a little trickier than plants because it's more difficult to, you can't really do tissue culture so much in plants. So what you have to work with is essentially plant embryos, okay? And you somehow have to get the gene that you want to move into the cells of the embryo. So unlike plants, where again you can just take a cluster of cells from the stem or leaf or whatever, here you have to specifically work with embryos. The vector that's often used to try to move the genes is a virus, what's called the SV40 virus. And so this virus, and this is just looking at, this is supposed to represent the genome of this virus. And so what you want to do is get your gene of interest into the viral genome. Then you've got your embryo, this cluster of cells. You expose it to the virus, and viruses normally function by basically injecting DNA into host cells, and so that's what you want the virus to do in this case, to get your gene of interest into your embryonic cells. If the process is successful, your embryo will develop into an organism that contains and expresses the gene you want it to move. Now, these steps here are by no means foolproof, and so it takes a lot of embryos and a lot of exposing the virus until you finally get embryos that successfully develop into these transgenic organisms. These are zebrafish that have been genetically modified. They're transgenic. They basically contain genes, I believe, from jellyfish that make them sort of have these different colors. Zebrafish are normally not this colorful. And I think I was reading, this is the only transgenic organism that is readily available for people to go out and buy and have in your fish tank, for example. There are other transgenic organisms, but they're more restricted in who can use them. There are some experiments being done with cattle and sheep and goats to have genes spliced into them such that these organisms will produce certain compounds in their milk that they would not normally produce. I don't believe that these types of animals are widespread in big herds yet, but this is a area of research. Now, another thing, animal cloning. Now, with plants, it's easy to clone a plant. You can often just cut off a little branch of something, stick it in some water, and you can get some roots to sprout, and you have essentially cloned that plant. Animals are much more tricky. What you have to do with them is essentially you have to get an egg cell, which again is sort of the ultimate stem cell. It's completely undifferentiated. You basically remove its nucleus. You take a nucleus from a cell from the organism you want to clone, and that nucleus can't just come from any kind of cell. You typically have to find a stem cell within that organism, because if you just took it from a skin cell, for example, in that skin cell, a lot of the genes have been turned off. And so when you attempt to grow an embryo from it, it won't grow properly. So you tend to find relatively undifferentiated cells from this organism you want to clone. You take its nucleus out, you put it into the egg from your organism that's donating the egg. If it's successful, it will begin to divide into an embryo. You can implant that embryo in a foster mother. And if everything goes well, that foster mother will give birth to a clone of this animal here using an egg from a different animal. This process has been done successfully. It took a lot of research to get the technique down, but a sheep was the first mammal to be cloned. There are now dogs and cats and presumably other types of mammals that have been cloned now. And in theory, it's possible to do it in people, although it's pretty much illegal just about everywhere that I know of or that I've heard of to do that, to clone a person. It's considered highly unethical. And of course, some people disagree that we should do it with any kind of animal. But there are, I believe it's at Texas A&M University, there are some researchers there. There's a lab that if you want to get your dog or cat cloned, they will do it for you. You just have to provide DNA samples from your dog or cat you want to clone. They will do this procedure. It's not cheap. In fact, I think I've heard it costs many tens of thousands of dollars to have this done, so you have to really, really like your dog or cat. And I'll show you in class some pictures and examples of this. And when you do it, The clone is not almost it's not necessarily exactly alike the one you're trying to clone, um, because gene expression in clones can sometimes be different than in the organism you're trying to clone. Um, but we'll talk about that in class. Okay, that's it for section 2 of chapter 15.